Hello biology buddies, and welcome to lesson 7 from unit B1, cell biology. In the previous lesson, lesson 6, we talked about animal stem cells. In this lesson, we're going to be talking about plant stem cells. We're going to revisit the idea of stem cells and differentiation, but then talk quite a bit more about cloning as well. So on that note, here's what we're going to do. Just like in lesson 6, we're going to talk about what a stem cell is and the process of differentiation. Remember, differentiation can appear on the exam or your textbook as cell specialization. For the purposes of GCSE, they're going to mean the same thing. We're going to talk about where stem cells are found in plants. We're going to explain how plants are cloned, and then we're going to talk about the benefits of the cloning. Cloning comes up quite a bit on the GCSE spec. So cloning comes up in unit B1, which is where we are. It also comes up in unit B3 if you're doing the separate biology, when we talk about monoclonal antibodies. And then last but not least, it comes up quite a bit in unit B6. So it can come up on paper 1 and paper 2 for combined science students and separate science students. So really, really important that you have those definitions of cloning down. What is a stem cell? A stem cell, as we saw last lesson, is an unspecialized cell that can divide and differentiate into specialized cells. It's going to divide through a process called mitosis, which we'll be examining in the next lesson. Lesson 8 is all about mitosis and the cell cycle. And differentiating into specialized cells. We've talked about some specialized cells in plants. We talked about the root hair cell in a previous lesson. We've talked about palisade mesophyll cells. Well, the palisade mesophyll, which contains the palisade cells, and then also the xylem and the phloem. These all come up in unit B1 and B2. Well, palisade cells really just come up in unit B2, but everything else comes up in 1 and 2. Differentiation is similar in plant cells as to how it happens in animal cells. So you start with your stem cell, your cell that can become other types of cells, and it will differentiate into a specialized cell, that root hair cell, palisade cell, xylem, phloem, etc. When it is differentiating, when it is specializing or turning into a specialized cells, what's happening is there's different genes that are being turned on or off, which is then going to, as you'll see in unit B6, instruct for different proteins to be produced or not produced. And that's going to change the structure, so not just the shape, but also the organelles that are within it, of the cell, which is going to change its function. Remember, in biology, whenever we talk about structure, we always should be talking about function as well. And if you know the structure of a cell, you know what sort of organelles it has, you can usually take a pretty good guess at what its function is going to be. Conversely, if you know the function of a cell, you can work out what its structure is going to be. So just a reminder from when we did the lesson on specialized cells, a root hair cell is going to be in the root, so it's not going to be exposed to the sun. So right away we know that it's not going to do photosynthesis, which means in terms of its structure, it's not going to have chloroplasts. Meristem is a new concept for plant stem cells. In animal stem cells, we talked about embryonic, we talked about adult. We don't need to worry about that sort of distinction in plants. Instead, we have something called the meristem. And the meristem is a tissue, so it's made up of a whole bunch of undifferentiated cells, so essentially stem cells. The meristem rapidly divides, so you get new meristem constantly, and that division is going to happen through mitosis. And the meristem is primarily found in the shoot tip and the root tip. And that's where you're going to get most of the growth of the plant, right? It's going to grow up through the making the shoots larger, and then as it grows, it's going to need more mineral ions from the soil and more water, so the roots are also going to grow. So that's where you have most of the meristem. That meristem can differentiate into any specialized plant cell. So for example, xylem, phloem, root hair cells, etc. And the xylem and the phloem, as we saw, are the transport tissues of the plant. The xylem transports water, and the phloem transports sugars. So water and the mineral ions that are dissolved in them, and the sugars. So as the plant grows, it's going to need more xylem to get the water to where it needs to go, and it's going to need more phloem to move the sugars around where it needs to grow. Sometimes people get a little bored with the plant stuff. That's not uncommon at GCSE or A-level or even university. mainly because we don't see plants doing a lot of stuff. But if you think about it, plants are actually pretty incredible forms of life, and they're very, very different to us as animals. So I would like you to, when you get the chance the next time you go outside, appreciate a plant, look at some grass, look at a tree, etc., and think about the ways that they're different to humans. And a lot of that has to do with their meristem. By that, what I mean is a plant will continue growing as long as it has the space and the resources to do that. Humans do not keep growing their entire lives. Take a look at a tree and think about how big it is, and it came from a single seed. There's no cap in terms of the internal programming of the plant to how big it should get. That tree can just keep growing and growing and growing. The reason, if you have a garden, that you need to go out there and keep cutting the grass all the time is that that grass, as long as it's alive, will continue to grow and grow and grow. And one of the main reasons for that is in the shoot tip, you have the meristem. So it's going to continue to be able to make new plant. It's almost as if the plant is regenerating when you cut it, as long as you don't kill it. And then in terms of the trees and other plants that just continue to grow and grow and grow, that's because the meristem stays there. It keeps getting pushed forward, forward, forward in the shoot tip and the root tip. So you have this, in theory, unlimited growth. Really, you'll get limits due to space and resources such as sunlight, water, mineral ions from the soil, and things like that. But plants can continue to grow, and humans can't. It's probably for the best because you'd have to continue buying new clothes all the time, and you wouldn't be able to fit through doors probably after your 20th birthday. But it is a neat way that plants are different to humans. So, to get back to what we were talking about before, rather than our weird tangent about why you should appreciate plants and go and hug a tree the next time you get a chance, the meristem, when we talk about stem cells and unspecialized cells in plants, that's the big thing. As long as you remember where the meristem is and the fact that it can differentiate to become any type of plant cell, you're pretty much set. The rest of this lesson is going to be about cloning, how you can clone plants, and why cloning is done. So a clone, as we said, this comes up in Unit B1, B3, and B6. A clone is a genetically identical organism. It has the same DNA. It's going to have the same alleles as you'll see in Unit B6. Really, really important that you remember that definition. They love to ask it on both Paper 1 and Paper 2. It'll come up as a 1-mark question. What is a clone? All you need to write is genetically identical organism. If we think about it, there are natural clones and sort of man-made clones or human-made clones. A natural clone in terms of humans would be identical twins. They are genetically identical. They are clones of each other. When we talk about things like plants, you have strawberry plants that have runners. You have other plants like tubers and bulbs that can also produce clones. So plants are able to reproduce in a variety of different ways. And when we talk about Unit B6, you'll talk about the differences between sexual and asexual reproduction, and we'll talk there more about the different ways that plants can clone themselves. But if you've ever seen a strawberry plant, the little sort of, it looks almost like a stalk or a stem shooting out from the side, that's a runner, and the plant is looking for somewhere else where it can create a copy of itself. Spider plants have the same thing. Tubers will do it underground, bulbs will do it underground, but essentially they just break off a piece and create a clone of themselves. So humans have been cloning plants for a long, long, long time. Sometimes when we think about cloning, we think of science fiction. We think it's a recent thing. But when it comes to plants, humans have been cloning plants for hundreds and hundreds, if not thousands and thousands of years. And there's two main ways that you can clone plants, and both of these come up on the GCSE AQA specification. The first one is cuttings, and that's the one that humans have been doing for a long, long, long time. And then a more recent way of cloning plants is called cell or tissue culture. And what we're going to do here is go through the two different ways. Nice 3, 4, even 6-mark question on an exam. Talk about cloning in plants and in animals and the different ways that they're done. So cuttings. One way to clone a plant is you simply cut off a piece of the plant. That's why it's called a cutting. And if you cut off a piece of the plant that contains the meristem, then you are potentially going to be able to create a clone of that plant, because that meristem is essentially stem cells that can become any type of specialized plant cell. So as long as you put that meristem that you've cut out in an appropriate place, if you give it the hormones it's going to need, you give it the mineral ions it's going to need, you give it the the water it's going to need, you can grow an exact copy of the original plant from the cuttings. And humans have been taking advantage of this for a long, long time, especially with things like apple trees and things like that, where you want to, if you have a variety that you really like, you want to get more exactly like that, you can use cuttings to get identical plants, and therefore, provided they have the same environmental conditions, identical fruit or vegetables coming off. So the key here is you're cutting out the meristem, which means you're most likely cutting off part of the shoot tip. The meristem is made of unspecialized cells, which can make all of the specialized cells required for a new plant, and it often requires the use of plant hormones. This comes up in separate biology in Unit B5, and you'll hear about rooting powder and especially the hormone auxin. Okay, so something to think about for Unit B5 is we can link that back to plant cloning and cuttings, and this idea of using rooting powders that contain the hormone auxin. Obviously, you're using rooting powders to encourage that meristem to not only grow shoot, but also to part of it to differentiate and become the roots of the plant. The other way is called cell or tissue culture, the other way of plant cloning. And for this, you can actually take the meristem, and instead of having that big piece of tissue, you can break it up into smaller bits being as small as individual cells. So then you can make an entire plant out of, in theory, one plant cell that was taken from the meristem of that tissue. We need to remember that the meristem divides very rapidly. It's going to make copies of itself. It's going to make daughter cells through mitosis. So if you have that one meristem cell, you have that one unspecialized plant cell, it will start to copy itself. It will start to divide through the process of mitosis, make a whole bunch of unspecialized cells, and then those can differentiate to become the different parts of the plant. So you couldn't really do this in the past because there was no way of being able to separate the meristem into individual cells. People didn't even know what cells were. But now, with one piece of meristem, in theory, you can make loads and loads and loads of identical clone plants. You'll often see this referred to in gardening as micro-propagation. So the idea of growing new clones from very, very small bits. And you can make a large number of clones from a single plant. So again, you're using the meristem. If you're thinking plant cloning, if you're thinking plant stem cells, you need to have that word meristem ready to use. Whereas in cuttings, you use a big piece of meristem. For cell or tissue culture, what you're doing is breaking that meristem up into smaller and smaller bits, and each of those bits you can then use to grow a new plant. So there's the two ways of doing plant cloning. Again, that comes up on the paper 1 exam from B1 stuff, and it comes up on the paper 2 exam from B6, and even a little bit from B5 if you're doing separate biology. Okay, so a lot of effort here, a lot of time, hundreds if not thousands of years people have been cloning plants. Why would you want to clone plants? There's a lot of reasons, but there's two main ones that you're going to want to talk about on the exam. The first one is if you have a plant that shows desired characteristics. It could be disease resistant, so it's resistant against a disease that's common in that area. Yield, you're getting more of the plant or you're getting more fruit. Maybe if it's flowers, you're getting a certain color, something that is desirable and therefore that you can make money off of. So if you have a whole bunch of, let's say, apple trees, and one of the apple trees is producing these delicious apples that everybody in the world loves, well, why not make yourself a whole bunch of apple trees that are exact clones, genetic copies of that original one? Because then those are going to make really, really delicious apples too, and you can sell all of those. Maybe if it's flowers, maybe if it's wheat and it has disease resistance, then you just want copies of that wheat. You don't want to mess around and hope that some of your future varieties also have that disease resistance. You just want to have as many copies of that plant as possible, so you would do cloning. The other is more from a conservation perspective. So if there are endangered or rare plants, then what scientists and conservationists will do is clone that plant in order to have more copies of it. So two main reasons that people clone plants. The first is for commercial reasons. If you have a plant that is great and has a wonderful characteristic that you can make money off of, then you'd make even more money if you had more copies of that plant, and that's what a clone is. Secondly, if you find rare plants or you find endangered plants, then you can create more of them through cloning as well. That's it for the plant biology, the plant stem cell stuff. Remember, it'd be useful to compare what you've done and your notes you've taken against the stuff from from lesson 6, the animal stem cells. But what have we done? We've talked about what a stem cell is. It's an unspecialized cell that can turn into or differentiate or specialize into a specialized cell. And in plants, examples of our specialized cells are root hair cell, palisade cell, xylem, and phloem. Describe the process of differentiation. That's where the stem cell becomes a specialized cell by turning different genes on or off, which changes the shape and structure of the cell and therefore its function. Describe where stem cells are found in plants. They're found in the meristem tissue, which is in the shoot tip and the root tip of the plant primarily. Explain how plants are cloned. There's 2 different ways that we said. There's cuttings and there's cell or tissue culture. Both of them involve harvesting the meristem from the plant that you want to clone. Explain the benefits of plant cloning. You can make more money if you have copies of a plant that has desired characteristics, a certain quality of fruit, a certain color of flower, some disease resistance, etc., etc. But also you can use it to preserve rare or endangered plants. So that's it, a nice short one. Be sure to fill out the form questions. Next lesson, we are going to have a look at the cell cycle, so essentially the life of a cell, and mitosis, how cells divide to make new genetically identical cells. Thank you so much. I hope you and your friends and your family are safe and continue to do so. Until next time, bye-bye.